Thanks everyone for joining today's Adelaide Biomed City Minireview. I'm Peter O'Loughlin from SA Pathology, and at SA Pathology we're very proud to bring you two very stimulating presentations on COVID testing at SA Pathology that really show the rapid mobilisation of existing expertise to develop and deliver a world-class testing service that has been central contributor to the control of the pandemic here in South Australia, and we've done well. So I think we are very proud, and we've got reason to be proud of the people you're going to listen to today. Our first speaker today is Dr. Ivan Bastian. Ivan is the Clinical Director of Microbiology and Infectious Diseases at SA Pathology. He has worked as a clinical microbiologist and researcher for more than 20 years. And Ivan has also established a WHO supranational TB reference laboratory here in South Australia. The title of Ivan's presentation today is COVID-19, a Laboratory Perspective. Thanks, Ivan.
So thank you for the opportunity to speak today. I'll be speaking, as Peter said, on our laboratory perspective on COVID-19. So what I'm going to quickly go through is just the amount of workload that we endured and achieved, the diagnostics, and I'll just mention how little was actually done by the commercial NAAT testing. So we actually had some commercial company tests, a Gene Expert, which allowed us to do a rapid 1-hour test, and also we used it for supplementary testing. And we also had some commercial serology tests, which we're using at the moment. But as I'll highlight, it's really the in-house assay that achieved everything for us. And finally, I'll finish off just looking to the future a bit in a brave new world once we get vaccinations and things out there, and where I see universities and others could be heading. So just to highlight the testing numbers, and this is right up until yesterday. So we've done 902,000 tests at SA Pathology, and 88.9% of those have been done by the in-house nucleic acid amplification testing that I'll highlight in this presentation. And you'll see basically that SA Pathology has done the huge majority of the testing in South Australia overall. And also, just to put it in context, how well we've done compared to interstate, you'll see that this is the green line. This is taken from a website if you want to look it up. The green line is South Australia, and we compete for tests per 100,000 population with New South Wales and Victoria, even though they've had their outbreaks. And I'd highlight, it's not really visible here, but at the really important times back in March, April, May, we were actually leading the pack as far as tests per 100,000 population for SARS-CoV-2, because it was actually incorporated into our routine respiratory viral PCR test. So if you had an influenza test or a respiratory syncytial virus test in South Australia, your doctor, your GP, as a bonus, got the COVID test. So we were actually screening in a way continually for SARS-CoV-2 from the start of the pandemic. And this just shows, this is going back over now, 3 years or so of data. I'll highlight that this dotted black line is the tests per week, and that's down here. So we're doing less than something around about 1,000 tests per week, or 5,000 tests per week, 10,000 tests per week at various stages over the last few years. Here, less than 1,000. We've been doing 1,000 today, and in fact we hit 12,700 tests per day in the middle of the parafield cluster. So what was being done in a week is what we've been doing in a day during the SARS-CoV-2 pandemic. So well, lots of initiatives that achieved that turnaround time, and I'll highlight in-house PCR. But SA Pathology also had the first drive-through clinics in Australia and the second in the world, had lots of extra collection data entry staff. There are great problems in getting swabs, reagents and consumables, so that took a lot of initiatives there. We did get in 3 commercial platforms, so described. We actually launched an SMSing platform, so results were SMS directly to patients as soon as they came off the system. And Chuan is going to talk about the whole genome sequencing. However, I'll highlight that it was really the in-house PCR that achieved this. And I'm probably the last person who should actually be speaking about this to you. I'd hope Mark Tara, this person who actually developed this PCR and the whole testing platform. He recently won an SA Health Award for that. And Jeff Higgins and Trish Hazy were the scientists and medical leads for the virology director that achieved that. So really all of these people are far more involved in this testing platform. So what was this in-house NAAT test? It was actually an in-house assay that was developed by Mark and Jeff and Trish and others over the last decade. So this was really 10 to 15 years in the making. This platform is what we've used for that period of time to do our respiratory viral PCR testing normally, our enteric testing normally, our MRSA testing normally, our Group B strep testing normally, and that we had this in place which could be upscaled and could have the primers and probes and things put in for SARS-CoV-2, just put into this normal testing in-house platform that gave us the capability to do this and also was scalable. So basically the in-house NAAT is a manual processing and inactivation of the specimens as they come. They get automated, pipetted into 96-well plates. They then go into a nucleic acid extraction. I'd highlight there's two of these. We were using the Roche system, but Roche couldn't keep up with reagents, and we actually were able to go back to the old MagMax that we'd used somewhere between 3 to 5 years ago, and could actually go backwards, and we could actually get generic reagents for this system. So we were able to do nucleic acid extractions, and then after we've got the nucleic acid extracted, it then goes into the samples get pipetted into 384-well plates and a master mix added to do the cyclers on thermal cyclers. So this is all an in-house system, all overseen by software packages run by the staff with a turnaround time of about 5 hours. I'll highlight this has been the workhorse. It's performed more than 85% of our testing. And the beauty of this is it's independent of the test development or supply chains of commercial companies, whereas many other labs, some in Australia, but certainly in the US and UK and other places, they were hanging out for Roche or whoever to make the assays and also to keep up with the reagent supplies, which was a struggle. We were able to have independent test development and the supply chains of generic reagents. We've been able to do this because I've highlighted this has been our routine testing platform for more than a decade, and it's been scalable. The SARS-CoV-2 sequence was released, I remember, on the 14th of January. I got it as a tweet. I emailed it to Mark Tara, who was already over this, and within 2 weeks we actually had a SARS-CoV-2 PCR in place as part of our routine testing. And that's been evolved over time. It does need some requirements. It needs investment in expert scientists and machinery. There's continuous in-house computing, verification and QC, and we do have to keep up with some extraction reagents and Hamilton tips, but it's allowed us to keep going and be basically independent of commercial companies. So where do I see this going in the future? One lesson I learned during the outbreak was that we need in-country manufacturing. We didn't actually make swabs in Australia until halfway through this pandemic. So we've been involved with a company in Sydney for evaluation of 3D printed swabs. And in fact, some states are now using those 3D printed swabs that have been modified for saliva testing. I'll also say there's been other initiatives with universities and others to develop a plasticware for that MagMax machine, and also the reagents for the nucleic acid extractions needs magnetic beads. They were made overseas. They're actually off patent now. And so SA Pathology, others, Jeff Higgins and others have worked with companies and universities here in Adelaide to try and develop those reagents for the future so that we're even more self-sufficient. if you ask me what universities and others should be working on into the future, I think we've got to start preparing for a vaccinated population with occasional breakthroughs. So we're going to still need routine high volume NAAT and serology tests, though we've got serology tests now in place with Roche. So a lot of this is now in place, but if anyone's developing things, we need high volume, easy to perform tests for these things in the future. So I think that's the end of my presentation and dreaming of a post-COVID world on my little trailer sailor in the future. So thank you. Happy to answer any questions.
Thanks, Ivan. That was a fabulous presentation. It's quite impressive what your team's done. So thank you very much. I do have a kick-off question here from Doug Brooks. Is there any mRNA post-viral infection testing?
Not that I'm aware of. Chuan might be better placed on it. We're certainly looking at various ways of trying to see whether this is recent or past infection is one of the things that we've been looking at. But this is more in a research development area, but Chuan might be able to answer that better than I.
Yeah, that's a good question. So we certainly had a look at the use of subgenomic RNA detection previously, but some of the evidence that we have come across is that there's a shorter shedding period post-infection if you detect it with the subgenomic RNA. But what we have found is that with the you introduce complexity in terms of the interpretation of the test, and we have a good understanding of the viral dynamics, and also in combination with serology, we can often tell whether somebody is infectious or not. So we haven't actually needed to do the subgenomic RNA detection for that reason. And they have a better application in cell culture and that form of experiments, but in real life testing, we don't find it that particularly useful.
Just one question from me, that we don't have any other questions that coming through the Q&A function, but Ivan, do you think that the occasional breakthroughs that you're predicting might pose different types of challenge, diagnostic challenges in the future?
You're asking me to crystal ball gaze here, Peter. It's a little bit difficult, but I imagine so far none of the, well, it tells a bit of a lot. Generally speaking, most of the variants haven't affected our diagnostics, and we're keeping a view on the variants with the whole genome sequencing, which Chuan will attach, so that to make sure that our diagnostics aren't affected by any of this. So I think our diagnostics are well in place to check for these breakthroughs. Where it will be, I think, is important is to track these and for the whole genome sequence to see if there's any particular variant that's, you know, escaping from vaccination coverage, and that will be some of the challenges into the future.
Sure. Look, thank you very, very much, Ivan. That was very worthwhile. It was a fabulous story to tell. I think we'll now move on to our second speaker. That's Dr. Chuan Kok Lim. Chuan is a clinical virologist specialising in public health and epidemiology in the Microbiology and Infectious Diseases Directorate at SA Pathology. He's also an infectious diseases physician at the Department of Infectious Diseases at Royal Adelaide Hospital. Chuan has research interests in viral hepatitis, particularly HBV, and also stem cell monitoring of viral infections. Chuan's affiliated with the Viral Pathogenesis Research Lab at the School of Biological Sciences at the University of Adelaide. The title of Chuan's presentation today is Outbreak Investigations of SARS-CoV-2. I learned from Ivan's presentation not to call it SARS-CoV-2. SARS-CoV-2 using next generation sequencing. Thanks, Chuan.
Okay. Thanks for the ABMC Organising Committee for the invitation. I'll move straight into my presentation today, which is divided into 4 different parts. First of all, I'm going to give a brief overview of the role of the Public Health and Epidemiology Division of the laboratory, and then followed by the SARS-CoV-2 whole genome sequencing development And next, I'll highlight some of the important applications of whole genome sequencing on SARS-CoV-2. And then finally, the future development in the pathogen genomics in SA Pathology. And my main focus of this talk would be to illustrate our approach of whole genome sequencing development for public health purposes. So the Public Health and Epidemiology Laboratory is a division under the Microbiology and Infectious Diseases Department in SA Pathology, with the main function of providing disease surveillance for a wide range of different diseases for South Australia. Another important function is to develop methods to assist in hospital and community outbreak investigation. One example would be to provide genomic linkage for multi-resistant gram-negative bacterial outbreaks either in the hospital or neonatal intensive care units, just to name a few. So this slide highlights some of the ongoing threats that we have been monitoring over the years. And despite many of these infectious agents, many of them are viruses, never making it to Australia, SA Pathology has been gradually increasing our preparation for these diseases. In 2019 was plagued with the early outbreak of influenza, and then followed by SARS-CoV-2 pandemic at the end of the year. So in order to assist disease surveillance, we developed many methods and techniques over the years for typing of pathogens. And prior to 2018, this was done using a myriad of different techniques for bacteria, for viruses, for fungi. They're all very different. And many of these techniques are slow, cumbersome, labor intensive. In 2018, Ivan, along with our chief genomic scientist, Lex Leong, had invested time in moving some of these platforms into whole genome sequencing, and especially for bacteria. In early 2020, after 2 cases of COVID-19 were diagnosed in South Australia, we insisted that this technology needs to be developed in South Australia. So we were given 4 weeks to develop and validate these tests, and that represents an immense task because it involves a lot of things to be done, including, number one, we had to compare different extraction techniques and also different sequencing platforms to identify the best high throughput as well as most sensitive assay that allow us to perform direct sequencing from clinical samples. And also we need to develop our approach to whole genome sequencing, and that involves retrospective sequencing of all positive samples to establish a database for comparison, assessing intra-patient variation, sequencing of family clusters, sequencing of specimens from different body sites, and so on and so forth. And this was followed by development of bioinformatics pipeline, and also finally to validate the test, we actually needed to compare our genomic clusters with the epidemiology clusters to make sure that to compare their similarities or dissimilarities. So the protocol that we adopted involves a total nucleic extraction for clinical samples, followed by amplicon sequencing using multiplex PCR, and the sequencing library preparation was using Illumina termination protocol. So in South Australia, we have adopted a universal sequencing approach for all new cases, regardless of their epidemiologic information. So at the time, this was not a unified approach to sequencing SARS-CoV-2 in Australia. And this approach serves 2 purposes. Number one, it helps to build a database of SARS-CoV-2 sequences that we can actually compare with. And number 2, we can link cases with unknown chain of transmission. And in June 2020, as Australia was experiencing the large second wave of SARS-CoV-2 and every state was overwhelmed with testing and sequencing, it was very apparent to us that developing this technology locally was very, very important. And November 2020, our sequencing approach was vital in identifying the index case of a medical hotel associated outbreak that we call the parafill cluster. And so on the figure on the right-hand side here show the final genetic analysis of the B.1.36 lineage virus from the parafill cluster. And it shows that all the sequences in this parafill cluster show a very tight genomic clustering. So distinctly different from other B.1.36 viral sequences from around the world. And this information gave us confidence that the cluster diagnosed in the community and the COVID-19 case in the medical hotel return traveler was indeed identical. Here is another example of how SARS-CoV-2 whole genome sequencing helps resolve some unusual viral dynamics. And the chart on the left indicates the usual viral dynamics of SARS-CoV-2 in the upper respiratory tract specimens, and with most cases clearing the virus by 3 weeks from symptom onset. On the right, we have a patient A who was a healthcare worker that was diagnosed with COVID-19 3 months earlier, and then had 10 negative surveillance tests afterwards. 109 days later, since the initial diagnosis, this person returned to South Australia with a very strong positive real-time PCR for COVID-19, and whole genome sequencing showed 99% sequence coverage for this virus. And this actually raises a lot of questions of whether the result represent residual RNA shedding, or whether this person has been reinfected with a different variant of SARS-CoV-2. And whole genome sequencing and the rapid analysis of this case successfully link this case to an institutional outbreak interstate 3 months earlier, where this patient A was working at the time. Another example here shows that an unprecedented problem with SARS-CoV-2 diagnostic is that given the enormous number of testing performed, false positive result can occur even with the most specific tests. A late positive real-time PCR can pose a significant headache for the diagnostic lab and also public health officer. The yellow square indicates the sequence alignment of late positive SARS-CoV-2 sample with high crossing point values consistent with very low viral load. As you can see here, there are many gaps in between the sequences, and despite poor sequence coverage, the segments of the sequence match really well with different parts of the viral genome, suggesting that the real-time PCR was indeed detecting the real viral RNA fragments. So this gives us great confidence of our diagnostic assays. In late 2020, Southeast UK was experiencing a large outbreak of COVID-19, and preliminary analysis suggests an emerging variant from the lineage B.1.1.7, and this variant was termed the variant of concern, and it was linked to accumulation of a large number of mutations within a very short period of time with potentially higher viral load in patients. Since then, 5 different variants of concern have been described, some with risk of immune escape. And through the Clinical Disease Genomic Network, the CDGN, SA Pathology has initiated a working group for national surveillance of these outbreak lineages and mutations. Whole genome sequencing of the SARS-CoV-2 also allows us to perform functional modeling and monitoring for significant genomic changes in the S gene that encodes for the spike protein. And certain mutations in the receptor binding domain as an S gene can potentially result in vaccine failure of detection with certain real-time PCR platforms, and also even enhancement of infection. So in 2021, now that we have availability of vaccination, the work on SARS-CoV-2 genomic testing is likely to evolve, and with greater emphasis on surveillance of vaccine escape variants and also outbreak variants. Development of novel direct antivirals would probably need surveillance for drug-resistant mutations. So in addition, we will continue to develop whole genome sequencing of pathogens to expand its utility in both public health, but also for clinical applications. And one such example is the development of the pan-viral whole genome sequencing platforms with potential use in diagnosing meningoencephalitis or unknown cause, for one example, or infection in immunosuppressed individuals. And we will continue to expand our repertoire of testing in both bacterial and fungal genomics to identify causes of prosthetic device infection and antimicrobial disease surveillance. So this is our group of genomic scientists as well as a supervising microbiologist involved in the whole genome sequencing, with particular acknowledgement to Lex Leong, who has been instrumental in introducing some of these development in whole genome sequencing. I would like to acknowledge support from our executive, Tom Dodd, all the members of the Microbiology Infectious Diseases Department, and Genetics and Molecular Pathology, in particular, Ming Lin and also Rosalie Kenyon, and SA Health, CDCB, CDGN, and also MRFF. Thank you.
Thanks, Chuan. Again, that was an impressive demonstration of how quickly you were able to get this service up and running, and we're all very grateful for that. Perhaps I'll ask Chuan, do you want to comment on the sort of challenge that you might face in the future with an ever-growing range of variants, and how challenging will that be? Or do you think that there will be emergence of particular interests, and others that are controlled may fall away and no longer be of much interest at all.
Thanks, Peter. So the surveillance of the variant of concern is always a challenging thing. So firstly, the definition of the variant concern is quite fluid at the moment, and it's changing on a day-to-day basis because of emerging evidence. And so there's a lot of discussion at a national level on what defines a variant of concern, whether this is actually caused by mutations or whether this is actually caused by the lineage itself. So we suspect it's a combination of different things. So this is likely to evolve quite a lot. And this is definitely a challenge that we have to face, because with so much information, we have to continue to evolve and understand what is actually constitute a true variant of concern. And so post-vaccination, one of the main focus would be to monitor for spike protein mutations, and some of the mutations that have been linked to potential vaccine failure. So that will be our main goal, is to monitor along that direction.
You've got a lot of work left ahead of you, and I don't think this is going to be over in any hurry. So I don't have any other questions. I just...
Peter, it's Ivan speaking, if I could.
Yep.
I just sort of might highlight for people that SARS-CoV-2 has had a silver lining in that the whole genome sequencing has really got a very high profile and has led to funding for this activity to the benefit of not just SARS-CoV-2 but viruses and bacteria in general. So because the public health authorities have been, and the politicians have been pushing for they're wanting the sequences yesterday, so they realise they need to pay for it. So we've actually moved very quickly to actually get 3 MRFF grants have come through, which are providing staff consumables and equipment if it was necessary. The other thing that's pushed ahead with is that there is what's called AusTracker. I'm not sure, Chuan could probably elaborate further, but there is actually a national repository and single data analysis pipeline for doing these analyses, which has been really trailblazed by SARS-CoV-2. But now a similar national pipeline can be used for TB, Salmonella, Neisseria meningitis. All of these things are being discussed and with funding. So there has been a silver lining that's led to the burgeoning of whole genome sequencing in Australia, which had otherwise been languishing a little bit.
Oh, that's a good story. Thanks, Ivan. We are virtually out of time. I guess one of the objectives of these mini reviews is to highlight expertise and capability that exists on this campus, and to encourage new collaborations. And Ivan and Chuan are hunkered down very much with diagnostic testing at this point in time, and the pressures on that are a little bit difficult for them to predict coming into the future, but they're certainly interested in collaborations probably a bit further down the track at this point, but certainly I'm sure they'd love to hear of people who are interested, but understand if they can't immediately respond to your interest. But look, thank you everybody for joining us today. I think it's been a very good set of presentations, and I hope you join us again at this time next week. Thank you all.